Dear ladies and gentlemen, dear colleagues, fellow students, dear George Daley. It gives me a great pleasure to welcome you all to a new edition of our long-standing lecture series. My name is Michael Boutros. Together with my co-director, Friederike Nüssel, we are part of the Marsilius-Kolleg, the interdisciplinary Institute of Advanced Study at Heidelberg University. Before I introduce to you George Daley, I want to say a big thank you to Isabel Steinbach with the violin and Franz Wassermann on the grand piano. You set the perfect mood for this afternoon with Meditation from the opera Thais by Jules Massenet. We'll be hearing you again at the end of this event with the famous Hungarian dance number 5 in G minor by the legendary Johannes Brahms. Thank you both for kind of the great contributions. Since 2008, the Marsilius lectures have been held every semester here at the Alte Aula of our university. The list of outstanding speakers who have impressed us with their expertise, intellect, wit, and rhetorical skills is long. Over the years, distinguished speakers graced us with presentations that crossed disciplinary boundaries, addressed scientific innovation, and reflected on grand societal challenges. They have stimulated the interdisciplinary dialogue at the Marsilius-Kolleg with new ideas and impulses, as well as furthering the public communication of science at our university. Recent lectures, for example, have dealt with psychological construction of time by Frank Worrell, technological advancements in health research by Don Imber, political dimensions of emotionality by Ute Frevert, and hope, the basic principles of human orientation by Ingolf Dalfert. Today's speakers will follow his predecessor with ease. George Daley is an internationally renowned scientist in the area of stem cell biology and the development of novel stem cell-related therapies. In addition, he is keenly interested in the implication, as we will hear, of novel technologies on society and human health. And as his talk, Scientific Progress and the Need for Oversight will show how kind of these novel technologies and application influence society. George Daley's impressive career began majoring in philosophy as an undergraduate at Harvard University, where he studied Immanuel Kant in a course led by no other than the famous social contract theorist John Rawls. Both apparently made quite an impression on him, as George Daley has since called Kant perhaps the greatest philosopher of all time in terms of his contributions to ethics, moral philosophy, epistemology, and logic, and Rawls as one of the preeminent political and moral philosophers of the 20th century. George Daley's subsequent decision to switch to biology research, good for biology and medicine, maybe not as good for philosophy, during his sophomore year should not be regarded as an abandonment of the study of philosophy and ethics. On the contrary, by his own account, these early experiences have greatly influenced much of his later work, especially that which is concerned with the interface of biomedical science and ethics. In addition to his own interest for interdisciplinary research, crossing disciplinary boundaries was also encouraged by the Harvard-MIT program, Health Sciences and Technology, which George Daley joined after graduating from college in 1982. For his doctoral studies, he went to the MIT-affiliated Whitehead Institute for Biomedical Research in Cambridge, where he worked at the laboratory of Nobel Prize winner David Baltimore. And some of his benchmarks I have seen are also here today. He became interested in hematopoietic stem cell disorders and turned his PhD in 1989 for groundbreaking research into chronic myeloid leukemia. 2 years later, he obtained his MD from Harvard Medical School and went on to pursue his clinical training in internal medicine at Mass General Hospital, before working as a clinical fellow and maintaining an active clinical practice in hematology and oncology as Mass General and Boston Children's Hospital for over a decade. Additionally, in 1995, George Daley already started his own laboratory then at the Whitehead Institute. His research had major impact on understanding chronic myeloid leukemia, on how pluripotent stem cells can be differentiated into different cell types to understand underlying disease processes, to model human disease, and also how they can be used as therapeutic agents. He was one of the founding members of the Harvard Stem Cell Institute. He was promoted to associate professor in 2004 and served as the director of the pediatric stem cell transplantation program at the Dana-Farber Cancer Institute and Boston Children's Cancer and Blood Disorders Center from 2009 onwards. He was appointed full professor at Harvard Medical School, where he became the Robert Stranahan Professor of Pediatrics and Professor for Biological Chemistry and Molecular Pharmacology. Since 2017, he's the Dean of Harvard Medical School and the Carolyn Shields Walker Professor of Medicine. He's also a member of the board of the Broad Institute of Harvard and MIT. Unsurprisingly, George Daley's work has been recognized by his peers with many awards at the highest level. I just want to cite a few. He has been selected Howard Hughes Medical Institute investigator, served as president of the International Society of Stem Cell Research, and continues to serve as an elected member of various professional societies. One of the many prestigious awards George Daley has received over the years, just three to highlight. He won the Director's Pioneer Award of the NIH in 2004, the American Philosophical Society Judson Dayland Prize for his achievements in patient-oriented research, and the American Pediatric Society's Mead Johnson Award for its contribution. Let me end by expressing really our heartfelt gratitude that you came to Heidelberg and taking the time to spend with us at the Marsilius-Kolleg and at Heidelberg University. Thanks very much for accepting our invitation and for delivering the 33rd Marsilius lecture today.
Thank you very much for that kind and generous introduction. It's absolutely wonderful to be here. It's such a distinguished institution that I last visited in 1991 when I was the guest of Dr. Thomas Wirth, who is here, very close and dear colleague. I want to thank Drs. Boutros and Nüssel and also Tobias Just for their kind invitation and also very wonderful hospitality while we're here. So let me say I was very much inspired by the mission of the Marsilius-Kolleg to bring together thinkers from different fields to probe the intersection of science and other fields, especially as it relates to the ethical and social context of scientific technologies. Indeed, in my career over the last 25 years, I've committed a considerable amount of my time to encouraging the responsible conduct of science by scientists, and I will share some of this experience with you today. So let me start my conversation today going back to a book that I read in high school, but a book that really had a transformative effect on my career, and that book was Brave New World. This was a dystopian science fiction that was remarkably prescient because the science fiction written by Aldous Huxley in 1932 has become a reality. He predicted at that time that there would be IVF, in vitro fertilization, and he predicted that there would be designer babies. And indeed, both technologies are now available and continue to be much debated. And Huxley's admonition to us in 1932 was to anticipate the emerging technologies and choose wisely whether and how to employ them. And that admonition has been a guiding principle in many of my attempts to engage with bioethicists, legal experts, those involved in the ethical and legal and social implications of technologies at the interface of science. So I will speak about just a short range of the technologies that I have witnessed and engaged in my career. But I'll just give a list of the extraordinary emerging biotechnologies that are raising vexing ethical and societal concerns. Reproductive technologies are in the forefront. In vitro fertilization, long thought to be a widely accepted practice, is once again coming under duress in the United States, where certain locales are actually moving forth with the prospects of restriction. My laboratory in 2004 was one of the first to demonstrate the derivation of germ cells from embryonic stem cells. And indeed, over the past 20 years, we have actually been able to demonstrate the ability to derive mice from sperm and egg that are created entirely in the petri dish. And the biology of human in vitro gametogenesis is not far behind. There's been a wave of reports of the creation of artificial human embryos, where the reassociation of different stem cell types actually can reconstruct an embryo that looks and behaves quite similarly to a human embryo. And there is increasing exploration of the principles of ectogenesis, the ability to gestate organisms like the lamb outside of the womb. There has been a rise in consumer genomics, the ability of individuals to have their genome sequenced and to be able to use that information, perhaps in the context of gene editing. There's also gene drives and genetically modified organisms. The ability to influence genes through the technologies that I'll briefly touch upon today have the potential to modify the biosphere. Regenerative biology, the field of stem cells, offers the prospects of life extension, and various chemical strategies are bringing the anti-aging approach into prominence. Synthetic biology, the ability to literally synthesize genomes and create organisms out of chemicals in a lab, and to be able to produce engineered biological systems that are completely distinct from what nature has provided us. Human-animal chimeras, xenotransplantation. Recently, a genetically modified pig gave a kidney donation to a human patient at the Mass General Hospital. There is an enormous amount of interest in development in the ability to couple the human mind to machines, leading to the prospects that someday neural implants will allow for the control of bionic limbs and exoskeletons. And then finally, and I will speak about the revolution that is before us today, the revolution in artificial and augmented intelligence. Now, all of this really suggests that the scientific pace of discovery, the pace is extraordinary, and we frequently as scientists realize that the science gets well ahead of our ability as a community to regulate and define how these new technologies should be applied. And indeed, the rapid pace of science provokes skepticism and opposition, and we're always reacting with academic and societal deliberation on the responsible oversight and regulation happening after the fact. So one of the strategies that I have attempted to put into practice through some of my engagements on the ethical and legal aspects of science is to ask the question of how can we become more proactive? So I'm going to talk about 3 illustrative examples from my own personal experience: stem cell therapies and embryo gene editing, the recently reported curative therapy of sickle cell disease using CRISPR for somatic treatments, not germline. And then I will finish with the discussion and some, hopefully, demonstration, if our technical support structure works, of artificial intelligence in medicine. And then I will finish with a recent effort on the part of the National Academy of Medicine in the United States to come up with a framework for the anticipatory oversight of emerging technologies. So let me start with a little bit of history on stem cells and embryo gene editing. Stem cells have been used in the form of hematopoietic stem cell transplantation for the better part of 60 or 70 years, and is now the standard of care for the treatment of certain types of leukemias, lymphomas, and genetic diseases. But some years ago, there became quite an interest in a more universal stem cell, not a stem cell that was restricted to the blood, but a stem cell that was a seed for all tissues in the human body, so-called pluripotent stem cells. And these hold enormous promise as models for understanding human developmental biology, which would otherwise be a period that would be opaque to human experimentation. And as I'll show you, it's been used in disease modeling, it's increasingly being applied in drug discovery, and there is the vanguard of its use for cellular therapies. I was first introduced to this transformative technology as a graduate student in the 80s. In 1981, Martin Evans, depicted here on the top, was the first to isolate from the mouse a mouse embryonic stem cell, and this was work that led to a revolution in the creation of genetic models of disease using gene modification of mouse embryonic stem cells, work for which Dr. Evans won the Nobel Prize. Sometime during my graduate work, I was introduced to Dr. Evans and told that these embryonic stem cells could be used to study the development of early human organs, and I adopted that aspiration to use embryonic stem cells to study the development of the blood. I started my laboratory back at Whitehead Institute at MIT in 1995 with the prospect that I might be able to capture the biology of blood development in a petri dish. And I was waiting for the prospects of human embryonic stem cells. And indeed, just 3 years after I started my lab in 1998, Jamie Thomson announced the ability to derive human pluripotent stem cells, pluripotent embryonic stem cells, from human blastocysts. Now, these were embryos that were left over from in vitro fertilization efforts and would otherwise have been discarded as medical waste. But the fact that in the US especially, the principles applied to the human embryo are such that some in our community believe that they are the equivalents of human beings and should be protected, created a firestorm of debate. And indeed, here on the cover of the MIT Technology Review was a report of the biggest prize in biotechnology, a cell that could be used to grow any type of human replacement tissue, but most companies wouldn't enter the race because it was a biotech taboo. And this stimulated in the early 2000s a whole range of controversies about whether or not US scientists would be allowed to study embryonic stem cells. Now, just before the report of human embryonic stem cells was an equally transformational report, a stunning demonstration by Wilmot Campbell and colleagues that one could take the adult cell of a ewe, a sheep, and reprogram it through a process of nuclear transplantation into an egg and reactivate the developmental process and produce the famous clone of Daley the sheep. So the juxtaposition of these two breakthroughs, the isolation of human embryonic stem cells and the ability to create an embryo by somatic cell nuclear transplantation, provoked in my colleague Rudolf Jenisch and me the prospect of putting these two technologies together. And we together published a proof of principle, not for reproductive cloning, but rather for an alternative called therapeutic cloning. The idea that we could use this partnership of nuclear transfer and embryonic stem cell derivation to produce a cell from a patient. In this case, our patient was a mouse with an immune deficiency. And as you can see here, we were able to use the reprogramming to produce an embryonic stem cell line which carried the same gene defect as the mouse. We were able to repair that gene defect in the Petri dish, produce hematopoietic stem cells, and transplant and restore the immune function of the mouse. This was an extraordinary demonstration that there was a promise of stem cell science, but it got embroiled again in controversy because to apply this in the human would require creating a human embryo and then leading to its destruction, or what some feared was that by fostering this technology, we would somehow be promoting the prospect. So we were being opposed to do this kind of research because of the prospects of human cloning. Now, in fact, technology was once again the solution to this ethical conundrum. And it was a technology of somatic cell reprogramming. The ability to take a few genes that specified the function of embryonic stem cells, and by adding these reprogramming factors to a skin or blood cell, we could make an induced pluripotent stem cell, which, if it came from a patient with a genetic disease, could undergo gene repair, making a healthy stem cell, and then differentiated back into a specialized cell that could be transplanted into the patient. This is now actually in the clinic for the treatment of individuals with diabetes, with Parkinson's disease, and with blindness, and we anticipate in the coming years that we will start to see clinical effectiveness of these types of therapies. But all the way back in 2008, this was called the Breakthrough of the Year in Science magazine, and it led to the awarding of the Nobel Prize to the scientist who first reported this, Dr. Shinya Yamanaka. Now, it's true that somatic cell reprogramming did quiet the ethical debate about the use of human embryos in stem cell research, but in some sense the genie was out of the bottle. And the fact that human embryos became a subject of study, whether natural or artificial, kept on the debate stage the prospect that human embryos could be genetically modified and create designer babies, as originally anticipated by Aldous Huxley in Brave New World. Now, the restriction on that had to do with the inefficiency of genetic strategies for editing. But that all changed in 2012 with the publication from these investigators here of a revolutionary and highly efficient new tool for gene editing, the so-called CRISPR. This is a tool that allows for the design of a targeted nuclease to interfere with any particular DNA sequence simply by targeting any gene by designing a specific guide RNA. This took a process that might otherwise have involved high expertise and months, if not years, of design, and it reduced it to a mere set of days. So the prospect that human embryos and gene editing could bring together the prospect of Huxley's Brave New World led to enormous consternation among scientists, and together with a small band of individuals, including Jennifer Doudna, David Baltimore, Paul Berg, and others, we called for an international conversation about the use of human gene editing. And this led to a first international summit on human gene editing, which was held at the National Academies in Washington, D.C. in 2015. And in that report of the organizing committee, it was felt that the development of gene-edited babies would be irresponsible. Indeed, whatever your ethical concerns were, and they were debated, the fact that the technology had not been proven safe was enough to lead for a broad international prohibition against the practice of this type of genetic designing. Unfortunately, 3 years later, the actual editing of human embryos and the birth of gene-edited babies was announced by this Chinese researcher. Now that led to a worldwide outcry. He had clearly flouted the standards of international scientific prohibition. It wasn't clear that there was broad understanding of the government of his malfeasance, but it seems difficult to imagine that he could have marshaled all of the resources of in vitro fertilization and gene editing and not had it be made aware by a very, very large number of individuals. And indeed, the Chinese government ultimately found him guilty of scientific misconduct, and he did serve 3 years in prison. What was the response of the international scientific community? Well, the International Society of Stem Cell Research again produced a set of guidelines that articulated guidance over not only stem cell research, but all embryo manipulations. And indeed, that guidance called for a deliberative process that would responsible conduct of science according to those principles, transparency, international collaboration, public engagement. And this applied not just to the range of methodologies that were being applied at the time in stem cell science, but also anticipated the prospect of emerging technologies that we could not anticipate at the time. I participated in the authorship of guidelines in 2006, 2016, 2021, and there were several lessons that I learned during that long experience. One, each of the task forces that was assembled to write those guidelines involved international representation. And indeed, I would argue that a proactive international engagement is essential when one is talking about regulation and ethical use, especially of reproductive technologies. It also follows that these standards should be revisited and reevaluated frequently in the context of evolving social priorities. The guidelines that we wrote in 2006, while calling for an absolute prohibition against human reproductive cloning, nonetheless evolved in 2016 the subsequent development of somatic cell nuclear transplantation or therapeutic cloning. Had those standards not been revisited and reevaluated, we might not have seen the transformation that led to somatic reprogramming. And then it's clear that even anticipatory guidelines and global consensus will not preclude the work of rogue actors. And it's an important principle that we can aspire to global consensus around various ethical applications of emerging technologies, but we can never preclude the work of actors who want to act with malfeasance. Let me turn to then what we thought would be a less controversial application of gene editing. It turns out that it had its own set of challenges. So the first somatic therapy that we all anticipated would yield to this new CRISPR technology was the treatment of sickle cell disease, sickle cell anemia. This is a devastatingly painful condition that affects primarily individuals who trace their history to sub-Saharan Africa. It was first described in 1910, and it was the famous scientist Linus Pauling who in 1949 actually reported that this was a structural difference between the hemoglobins in a diseased individual and a normal patient. And in the 1950s, this was shown to be due to a single amino acid substitution in the hemoglobin chain, which we now know is due to a single point mutation, a single letter difference in the 3 billion pairs of DNA in the human genome. Indeed, it was Pauling who labeled sickle cell anemia the world's first molecular disease. And he anticipated that our ability to understand disease at the molecular level would actually convert medicine into a real science. And that was tremendously prescient. 74 years it took from the first description of sickle cell anemia to a cure. And that cure was announced by a partnership of CRISPR Therapeutics and Vertex Therapeutics back last year, involving a very invasive and toxic therapy, but one which actually changes the course of history for these patients. This involves a bone marrow transplantation and gene therapy where the stem cells are extracted from the patient's bone marrow. They are treated to either restore the expression of a normal hemoglobin or treated with a genetic CRISPR trick to turn back on the normal fetal form of hemoglobin. The fact that there was such a long time to get there shows how hard this is. But we now have the first CRISPR therapy for the first molecular disease, and I wrote a perspective on it in the New England Journal of Medicine, which was titled Welcoming the Era of Gene Editing in Medicine. And I do believe that this is going to lead to follow-on treatments for a vast number of genetic conditions. So with this in hand, we made as the topic of the third International Summit on Human Genome Editing a focus on the breakthrough CRISPR treatment for sickle cell disease. And in particular, what we focused on was the fact that this scientific triumph was at risk of becoming a public health failure. And the lessons were because even though we had this therapy, there were huge hurdles to its broader acceptance. The cost, priced at over $2 million for that single treatment. So, as I said, the major hurdles were cost and lots of other challenges to scalability. And that was most acute because the greatest need of this disease treatment is in sub-Saharan Africa, where there are very, very few places that could sustain an autologous bone marrow transplant. There is a solution, and that solution is being actively investigated, and that would be the solution of packaging the CRISPR editing machinery into a particle that could be injected intravenously for in vivo gene editing. It's very, very challenging, but it's something that is being actively pursued and funded by the National Institutes of Health and the Gates Foundation in the United States. And the target, if those of you, the aficionados, the BCL11A target of the CRISPR modification is now a validated target, which is stimulating quite a robust request for small molecule drugs. And importantly, one of the principal messages to emerge from the third International Summit on Human Gene Editing was the priority for technology developers to anticipate the need for global access. And you'll see that that principle returns as I talk about our strategy for a framework for overall governance. So let me finish, I hope, with the last some minutes, uninterrupted perhaps, talking about another revolution. It's an emerging technology which is enormously exciting because it has, again, the prospect of enormous benefit, but also the prospect of risk, necessitating a responsible type of regulatory oversight. So I'm going to demonstrate, how many in the audience here are using one of the large language model AI systems every day? quite a few, then I think you might be aware of some of its remarkable capabilities. I'm going to show you, for those who aren't using it every day, the extraordinary potential for its use in medicine. Dr. ChatGPT, I want your opinion on a medical case. It's a 75-year-old overweight gentleman with a history of mild hypertension who awoke in the middle of the night after a spicy meal with stomach discomfort, belching, diaphoresis or sweating, dizziness, nausea, and vomiting. What diagnoses should I entertain? Now, please be brief. List the top 3 only in English, but then repeat the rationale for these 3 choices in German.
Of course. The top 3 diagnoses to consider are 1, myocardial infarction, heart attack, 2, acute gastroenteritis, 3, peptic ulcer disease. Rationale in German: 1, Myokardinfarkt, Herzinfarkt, Brustschmerzen, Schwitzen, Übelkeit, typische Herzinfarktzeichen, besonders bei Bluthochdruck. 2, akute Gastroenteritis, Magenbeschwerden, Übelkeit, Erbrechen nach scharfem Essen, Zeichen einer Magen-Darm-Infektion. 3, peptische Ulkuskrankheit, Magenschmerzen nach dem Essen, Hinweis auf Magengeschwür.
Thank you, Dr. ChatGPT. Now, I don't speak German, so I leave it to you to determine whether that rationale was sound. I think it's extraordinary to imagine that any practitioner around the world could be empowered in such a way to be able to use something as simple as a ChatGPT on one's phone. I wish that the doctor who had cared for my father, whose case I just described, had had the cognitive assistance of ChatGPT. My father was misdiagnosed. He was diagnosed with stomach upset and sent home without proper therapy and evolved a heart attack and ended up in the intensive care unit. He did survive, but I think it taught me a very, very important lesson. The fact is that we know that medical errors, especially diagnostic errors, are rife in clinical medicine. In this study, diagnostic errors were found in 23% of hospitalized patients who were transferred to the intensive care unit, the sickest patients in the hospital. This was judged to contribute to harm in 17.8% and to death in 6.6%. So the prospect of AI-enhanced medicine offers the possibility of fewer cognitive errors, better diagnostic accuracy, and greater clinical efficiency, but there may also be a downside. Now, interestingly, Vinod Khosla, who is a legendary entrepreneur, the founder of Sun Microsystems, and now a very influential venture capitalist in Silicon Valley, he wrote in 2016 a prescient article called 20% Doctor Included, where he speculated that technology, especially AI, would replace 80% of what doctors do. He wrote that computers are much better than people at organizing, recalling, and synthesizing complex information and decisions. And it's now estimated that medical knowledge doubles every 3.5 days on Earth. It's impossible for any human to keep up with such a pace. He also snarkily responded, "This will result in far fewer mistakes and biases than a hotshot MD from Harvard," which I took a little bit personally when I read that. But the idea was that AI would allow for something he called upskilling. It would allow individuals to be able to practice at a much higher level of authority and expertise, and that would equalize and democratize the practice and delivery of medicine. So what makes up that 80% that technology is going to make up for? Well, Think about it. How many of you medics out there are consumed with notes and documentation? AI is going to allow a simple bot to listen in on your interaction with a patient and write your note for you. Administrative tasks that we get burdened with in the United States, filling prescriptions, coding, and billing, can all be done by computers. Keeping up to date with medical literature and research assistance, and then finally, and I believe most importantly, medical diagnosis and the ability to assist the physician in best practices. Now, the data are starting to accumulate. Machines can perform as well or even better than humans in complex clinical scenarios. Here at the top, in analyzing skin lesions and calling them melanotic or not, the machine learning model's accuracy was comparable to that of general practitioners. But in other settings, here in data produced from a German group, the deep learning strategies outperformed 87% of dermatologists in melanoma diagnosis, and interestingly, in this series of experiments, the combination of human and AI was best. So the lessons to be learned there is that we have a lot to learn. These AI models can enhance, but they need to be crafted in ways that clinicians can use them. So let me speak about the differences between the machine learning models that have been the standard, especially as applied to radiology, dermatology, pathology, ophthalmology, anything that involves imaging and pattern recognition. If you can feed an enormous amount of training data into a machine learning algorithm, the machine, the computer, will be able to read and see subtle differences far greater than any human. So for these applications that are purely diagnostic, I imagine that the machine learning will become the standard very quickly. But there's been a transformation. What I showed you was interacting with an AI that actually was conversational. I could actually speak and interrogate that AI the way I might interrogate an expert consultant on the medical ward. And that's because of the emergence of the principles of large language models. These are computer algorithms that were developed in order to recognize human language and reproduce human language. They're algorithms that are based on something called the transformer architecture and the attention principle. The attention principle is essentially weighing the importance of words in a sequence called tokens. Tokens can be individual letters or words or even in some cases small phrases. And essentially this is a model that predicts the next token. And these models are trained on massive amounts of text data, and they're fine-tuned for specific tasks, which enable them in an extraordinary way to understand the context and can generate responses and engage literally in meaningful conversations. For those of you who have not interacted with ChatGPT, I encourage you to do, if for no other reason than it's tremendously entertaining, and I'll show you some of that. So GPT is just one of many models. There's Claude, there's Gemini, there's a number that are evolving, but GPT stands for the Generative Pre-trained Transformer. So a few more examples of where AI is going to help us in the clinical setting. So those of you who read the New England Journal of Medicine, which is what we do on that side of the pond, as opposed to Lancet, which is typical on this side of the pond, you'll know that there are case studies from the Massachusetts General Hospital. Typically, a distinguished clinician is presented with the case of a very vexing presentation of a fascinoma or some kind of zebra that presented in the clinic. And the clinician in front of his peers has to go through a long differential diagnosis, hopefully getting to the right conclusion. So here we are. This large language model was tested on 300 challenging cases from the New England Journal of Medicine. And how do you think the large language model did? It outperformed the clinicians. But interestingly, again, the best was a clinician plus the large language model, which enabled a more comprehensive and higher quality differential diagnosis. So the large language models can help us as clinicians. But can large language models replace clinicians? Well, interestingly, the performance of these large language models on the US medical licensing exam shows that just out of the box, with no special training, no special fine-tuning, the earlier version of ChatGPT performed at or near the passing threshold of the US medical licensing exams without any specialized training. And interestingly, if you look across the spectrum of other types of large language models, and this axis is simply 3 years, we've gone right through the passing threshold so that the largest and most sophisticated model right now, MedPalm, passes the US medical licensing exam with an 85% performance, which is strikingly better than most American medical students. Fortunately, not yet better than Harvard medical students, but they're coming after us. And the fact is that I would say in a few years, these large language models will be able to get these almost 100% right. So, will these large language models replace the cognitive effects of the physician? It should augment the cognition of a physician. But will they ever replace the human side of medicine? No, of course not. Well, surely doctors are needed for caring, but in fact, when explored head-to-head, and this was answers that were provided on Reddit's Ask Docs forum, so these were actual human doctors responding to medical questions from patients. A panel of licensed healthcare professionals preferred the ChatGPT response to those given by real doctors 79% of the time, which I think is pretty startling. 45% of the ChatGPT answers were rated empathic or very empathic compared to just 5% of doctors. So Vinod Khosla predicted 80% of what physicians do is going to be taken over by AI. The caring part may not be purely left to us. But let's say I believe that actually in the way that AI is supporting our diagnostic potential, it's also likely to make us more empathic. Now, what are the limitations? So this has been widely reported that these models make things up. If they don't know the answer, they simply make it up. So I'm going to show you, this is just something I did. Simple, and I invite you, it's sort of fun, the next time you're at a cocktail party, ask your friend to pull out their ChatGPT and say, write a biography of me. So a 100-word biography of George Daley, Dean of Harvard Medical School, indicating time of birth, hometown, blah, blah. So George Daley, born August 10th, the Dean of Harvard Medical School. You can read this. Okay. Looks pretty good. I wasn't born on August 10th. I was born on November 13th, 1960. I wasn't born in New York City. I was born in the small town of Catskill, New York. Oh, boy, I've received a few prestigious awards, but not that one. I am married to Amy Edmondson, but she spells her name with a D. And I would have loved to have had more kids. We have 2, not 3. So literally, they're still making errors. They are getting a lot better. And I can tell you, just in the 2 years that I've been using this platform, the error rates go down. And with specialized fine-tuning and training, the errors will go down further. So at what point does the cognitive benefit and becomes so much greater than the clinician, and the error rate much less than the clinician, do we turn over the responsibility for diagnostic work to the large language models? Now, of course, until that time, we have to be wary that hallucinations can misinform, can waste resources, or potentially result in dangerous health outcomes. And this represents an enormous ethical challenge. And as we think about who is responsible, where is the liability in the system? if a physician gives over to an AI. There is bias in training data, that if the training data is biased, the bias will be embedded within the algorithm. And there is the worry that AI will exacerbate disparities. The other point, and a point I think well taken, is that amongst the disparities are the very disparate advantages that are accruing to a very small number of high-resource tech companies. So this has prompted international action for regulation, and I should say that the European Commission has been particularly aggressive in this regard, and they have actually been the first to come out with regulatory guidance, a risk-based approach. The goal is that we want to make sure that the community can trust what AI has to offer so that we can maximize the benefit and minimize the risk. And they've assigned this risk-based approach, and I can allow you to read the notion of how we would approach a high-risk intervention. In the United States, we have not taken legislative approaches, but rather there's been executive orders from the president, and we have started, here is a Harvard Medical School-sponsored international symposium on responsible AI for social and ethical healthcare. So we have a ways to go. The technology is emerging, it's powerful, we still need to know how to use it. So I'm going to leave you with a few resources on AI and AI in medicine. These are very trusted books, they're very quick reads. My colleague Zach Kahane, together with Peter Lee, the head of Microsoft Research, and Kerry Goldberg, a journalist, wrote this wonderful book, The AI Revolution in Medicine. Henry Kissinger, together with Eric Schmidt, the former CEO and chairman of Google, together with Dan Huttenlocher, the head of the Schwarzman College of Computing at MIT, wrote The Age of AI. And there are journals like Lancet Digital Health and New England Journal AI that stand to filter for all of us the emerging technology of AI in medicine. So the strategy that we've finally sort of happened upon was I was fortunate to co-chair a committee at the National Academy of Sciences in Washington, D.C., the Committee on Emerging Science, Technology, and Innovation. And this was a committee that acknowledged that the benefits of scientific advancements are enormous, but so can be the risks. And so we decided that we would try to establish a framework of anticipatory governance. All too often, the science is running ahead of the ability to regulate. And so here was a framework where we imagined we could establish imperatives that any new technology and any new technologist and innovator should have the humility to subject themselves to public engagement, to the peer review and receptivity to oversight, to transparency, to freedom from conflicts of interest, and the application of storied principles of social justice. This framework that we proposed from this committee was a deliberative process, not a legislative one, and it's important, the distinction. Because guidelines are not laws. They're more easily modified as the science evolves and as the societal acceptance evolves. And so this CESTI framework was put forth as kind of living constitution, a set of principles, aspirational goals, and a deliberative process to live by. It was written up and followed by a special consensus study, which was published and is available on the website for the National Academy of Medicine. And interestingly, and back to that original principle that I articulated that's so relevant to the sickle cell therapy, how do we transform these scientific triumphs into public health triumphs? We need to ensure that the innovators pay attention to global access and equity early in the development of their technologies. That is, for me, one of our grandest responsibilities as scientists, as innovators. Now let me, in the last moment, change to something that I think you'll all appreciate, that ChatGPT is still in its infancy, and I would say for any applications in medicine, there still needs to be a human in the loop. But that doesn't mean that ChatGPT isn't enormously helpful for a variety of other purposes. ChatGPT, I'm a wine lover, but know relatively little about matching German cuisine and German wines. What dish should I try here in Heidelberg, and with what wine?
For a delicious experience in Heidelberg, try a traditional dish like sauerbraten, which is a pot roast typically marinated in a mix of vinegar, water, and a variety of spices. Pair it with a Spätburgunder, which is the German name for Pinot Noir. The wine's light body and bright acidity complement the rich flavors of the sauerbraten perfectly. Enjoy your meal.
Okay. All right, so I leave you with that. We have a technology that at least can help you order wine in a restaurant. Thank you very much for your attention.
So I'm awaiting you here to say thank you for this wonderful lecture, especially also for the historical part for people like me who don't know this and don't have to do with this all the time, but also for the wonderful and balanced exploration of AI, its benefits and its downsides, and also the imperatives. I think there is the Kantian at work. Thank you very much for this wonderful lecture, and I'm waiting here for you to fulfill a small ritual, which is to award you with the Marsilius medal. It's metal, it's not depending on any device anymore. Thank you also for your patience with this. And it goes with a certificate, which I will read out to you now. Oops. The Marsilius medal is presented to Professor George Q. Daley by the Marsilius-Kolleg of Heidelberg University for his lecture on the topic scientific progress and the need for oversight, and his contribution to the dialogue between scholarly cultures. It's signed by Michael Boutros and myself, July 1st. Thank you.
Thank you very, very much.
Here comes the medal.
I will treasure it. I will treasure it. Thank you. Thank you very much.